the next section in chapter 6 and 7 is about biotechnology. So what this section is going to be focusing on is how we've taken the information on how we use the central dogma, that information on how we go from a gene, DNA, to mRNA, then mRNA to things like proteins. So biotechnology is using technology to modify organisms, cells, and their molecules to achieve practical benefits of some sort. This can be anything from getting an organism to make a product like a medication for us, or improving crop yields or where a crop can grow, the nutrition of it, or even possibly in the future altering our own DNA to possibly cure a disease or something like that. So we're going to be looking at those different types of applications of biotechnology. Now, the thing is that biotechnology has something in common, and that is the processes that are used for biotechnology applications. So the first thing that you need to do with any sort of biotechnology is find the gene and the DNA of the organism that you're wanting to use. So let's say you find a gene in, humans to be able to make insulin, right? You are going to go in, find that gene and cut it out because you only want to work with that gene. Then you're going to copy it because just having one copy of that gene is not going to be enough. You're going to need hundreds, thousands, maybe millions of copies in order to work with it, depending on what kind of biotechnology you're going to work with. A lot of biotechnology then requires inserting that DNA into some sort of organism like a bacteria or into a virus in order for that virus to be able to transfer that genetic information. So, depends on what the application is, on what kind of organism, bacteria, virus, and so on that might be used. And then in some types of biotechnology, they actually are going to then grow separate colonies of these bacteria and viruses with different donor DNA. So maybe one group are going to be growing, insulin, another group would be growing, hemoglobin proteins, and so on. So the thing is that not all of them are going to use all of these different steps, but all of them do at least start with finding DNA of interest and generally making copies of that so you have enough to be able to do the different experiments that you'd like to. Now, the one kind of biotechnology that a lot of people have heard of in the news is genetic engineering. So genetic engineering is specifically adding, deleting, or transplanting genes from one organism to another. So we can take genes out or put genes in of a specific organism. So let's say in that case, we're working with like wheat. We haven't done this to wheat yet, but we could do this where we could exchange, genes between wheat strains, or we can actually transplant genes from one organism to another. So for example, we've got papayas here. Papayas in Hawaii almost went extinct because of a disease called ring spot disease. And we genetically engineered these with genes from other organisms to protect them from that ring spot. So they actually inserted genes from other organisms to help protect them. So we can do this in agriculture. We'll talk about some different ways we've done that. We can use it for human health. So you can see the gene chip here. So that's how some of the companies who do your DNA readings, if you've done the swabs or the spits and sent them out to get your genetic ancestry and stuff, that's what they're using there. But we can also use it for other things like treating diseases and possibly screening for different diseases, as well as in the future, hopefully, treating genetic diseases. And then we'll take a look at some different forensic science sections that we can use some genetic engineering for as well. So one of the newest biotechnology tools you may have heard of is called CRISPR. CRISPR is a new system for editing DNA. So previously we had a longer process that wasn't as precise for going in and cutting out these genes. But CRISPR has the potential to revolutionize medicine as well as other parts of the genetic engineering field and biotechnology as a whole. Because what CRISPR does is it allows us to modify almost any gene in any organism with incredible precision, but also efficiency. It's much, much more efficient than any of our other methods that we have so far. And this mechanism of CRISPR actually was discovered in bacteria and is found in almost half the bacteria in the world, because it's used to record encounters with viruses. So that way they can protect themselves against future infections. And so that's what they use it for, is to record this information and then use it to cut out viral infection information in the future. So how does CRISPR itself work? So what they do with CRISPR is it's part of this biotechnology, genetic engineering field. They're going to take a DNA sequence that they're interested in. So they find a section of DNA in, say, an organism that they want to work on. And the researchers make an RNA guide. So they make an RNA that matches one side of this DNA, right? It can match either side, but as long as it matches that gene, the RNA guide will be able to be used. So what are they going to use that RNA guide for? Well, they put that RNA sequence into what we call a plasmid. A plasmid is just a circular section of DNA that we can give to bacteria. And so this is the CRISPR plasmid. You give it that RNA sequence, and then you also have in here part of what was CRISPR, and this is what we call the Cas9 enzyme sequence. And so this is actually used to target the cells along with the RNA sequence to do this work for CRISPR, okay? So we're going to use this enzyme sequence and this RNA sequence. It's going to get introduced into the cell that we're working on. And once it gets in there, the RNA is going to take Cas9, so the Cas9 that gets made, to right where it needs to go on the DNA. Because remember, this RNA sequence matches the gene that we're looking for. So it's going to go there, and Cas9 is an enzyme that cuts. It's like molecular scissors. And so it's going to cut that DNA sequence out of the whole section of DNA. So it'll cut the gene out that we're looking at. And then we can have another section be pasted in, a new gene sequence be pasted in that either repairs or alters that host cell's DNA. And so this is actually one of the new mechanisms we're looking at for hopefully addressing some genetic diseases, because CRISPR is much more efficient at doing this cutting and replacing of genes. So we could possibly replace some faulty defective genes and possibly cure some genetic diseases that we have no treatments or no good treatments for now. And so this is how CRISPR works. And what can we possibly do with CRISPR? Because there's tons of work being done with CRISPR right now. This has allowed a lot of research to happen very quickly. So what is some potential of CRISPR? So CRISPR is actually being worked on to alter mosquito DNA so that they can no longer be infected by malaria, which is kind of cool. So they're giving mosquitoes malaria protection. And that way malaria spread will get stopped. Before this, what we were doing is basically having to try different things like killing out the mosquitoes in an area, or introducing sterile sterile males to mate with females so they wouldn't produce as many offspring. But if we can actually give them protection against malaria so they can't get infected, that could help us eliminate malaria in an area. We're also doing human trials where you have these CRISPR altered white blood cells, this blue guy right here, that are being programmed to fight cancer cells. So they only attack cancer cells and not regular cells. So these guys could be programmed to eventually be used as a cancer treatment. We have also been using CRISPR to engineer, I know this sounds funny, but engineer dogs with twice as much muscle as a normal dog. The idea here is possibly using this for other treatments in the future. We've also been able to use this for inactivating the immune system in tissues from pigs. And I know you're probably thinking, okay, so what does that have to do with anything? Well, pig tissue is actually one of the closest ones to human tissue that we can use for transplants. And so they were able to actually work on using CRISPR to edit 16 genes at the same time that could allow us to possibly now use tissues that were pig tissues as human transplant tissue. For specific purposes, and makes it now safe for us to use those tissues. So the other things are things like modeling human disease in cell lines without having to track down the cells that are already done. There are lots and lots and lots of different things that are being worked on. These are just a few of the different projects that are being worked on for using CRISPR. And the nice thing with CRISPR is that you can just edit within the genome of the organism. You don't even have to put information in to the organism from another one. CRISPR allows us to easily cut or add from the organisms themselves, because maybe it's just that we need to add in an extra copy of a gene or replace that broken copy. So what are the risks of CRISPR? So there are some risks of CRISPR. One is just about the legal issues. So there's still some legal issues about who invented it and who can profit from it. But that's hopefully going to get worked out soon. But lots of researchers and research labs are using it right now. There has been some talk about the ethical issues, mainly about using CRISPR for humans. We've been mainly focusing CRISPR on using it for other organisms like, plants, as far as crops and so forth, on like things like the mosquitoes for malaria. But there is the potential for using CRISPR to edit human embryos. So doing things like creating designer babies. On one hand, there is the argument of, well, maybe we could use this for, keeping genetic diseases from happening. But where do you stop that? If we do go into having this potential, because, if you've seen any of the, films like Gattaca and so forth, we could potentially edit these embryos to have specific traits. And then also the potential for editing germline cells. So going even a step farther back and editing the sperm and eggs themselves before they even fertilize. So there's some ethical issues that still need to be worked out and determined. And as we'll see with some of the other technologies we're going to talk about, it's difficult to predict the consequences of producing altered genes into these genomes, especially into natural populations. So for example, with those malaria mosquitoes, They are doing small trials to find out what happens when you mix these malaria altered mosquitoes in with a natural population to see, does it alter the genetic makeup of those organisms and cause problems in future generations. And that's something that we don't know. That is one of those risks as far as some of these types of technologies go, especially since CRISPR is so new. But CRISPR does have a lot of potential, and in the future, I'm sure you're going to see a lot about CRISPR in the news and in research about different new technologies coming out.